Yes, thank you very much for the introduction and also for the invitation today. I've been working as a child neurologist for the last 20 years, and rare diseases represent a very large proportion of the patients we see in pediatric neurology. And what I want to do today is to talk about one group of rare diseases I've been working on for the last 10 years, and at the same time to highlight some lessons I've learned from working on these conditions. And my original background is in early onset neuromuscular disorders, and that was what I worked on initially. But then, 10 years ago, I met a very unusual patient when I was the consultant on call for pediatric neurology. And this patient was one of the most severely affected children I've ever seen in my career. He had multiple problems, including structural brain abnormalities. He had a cardiomyopathy, a combined immune deficiency, and also cataracts and sensory neural deafness. And very sadly, he died at the age of 3 months, and I wanted to find a genetic answer for his family. But at the same time, I was also very much intrigued by the condition, because I thought whatever is wrong with this child must affect something biologically extremely fundamental. And although we didn't have an initial genetic diagnosis for his problem, we came up with a syndromic diagnosis. And the syndromic diagnosis was a condition called Vici syndrome, which was described in the 1980s by Carlo Dionisi Vici, a metabolic pediatrician in Rome, shown here with one of his patients. And at the time, there were only 8 cases reported with this condition worldwide. And we published a case report on this patient, and I think the fact that there are 18 co-authors on a case report, which is a personal record, illustrates how much expertise and what a multidisciplinary approach is actually needed when you look after a child with a rare disease. And we also wanted to report the muscle biopsy findings at the time, which were really intriguing because they suggested a very wide differential diagnosis of possible other conditions. The muscle biopsy had some similarities with early onset muscle disorders such as centronuclear myopathy, myotubular myopathy I have been interested in for a long time. But there were also other features reminiscent of glycogen storage disorders, of a lysosomal disorder, and of mitochondrial disorder. And there was a very wide differential diagnosis at the time, but the answer to the question, what is the genetic cause of this condition, was a completely different one. We did, after we published the case report, we got further cases, and that was at the time when whole exome sequencing was introduced into clinical diagnostics. And I was asked by my colleagues in the research lab if I had unresolved patients I wanted to have investigated by whole exome sequencing. And I thought Vici syndrome is such a well-defined condition that I was pretty certain that these patients would have the same genetic defect, which turned out to be the case. And we also included the original patients reported by Carlo Dionisi Vici in this study. And the gene at the time was not very well annotated, but it turned out to have a role in a process called autophagy, which is an essential intracellular pathway which is conserved throughout evolution. And the basic mechanism of autophagy is a process of digestion of intracellular components, in particular by the lysosome. And that is also the reason why these conditions have strong similarities, for example, with lysosomal storage disorders and other multisystem disease. And autophagy has 3 major roles. One role is to aid metabolic adaptation during times of increased metabolic demand. The second role is defense against infective organisms. And the third role is removal of defective proteins and organelles. And I don't think that these roles are equally important throughout evolution and throughout lifetime, because if you look at the first two roles, they are absolutely essential for the cell and the organism to survive. But the third role becomes more important once the cell and the organism ages, because as the organism ages, defective proteins accumulate and they will have to be removed from the system. And autophagy can be separated by target and by mode. I have highlighted in blue certain targets such as neuronal sheaths, protein aggregates, and the mitochondria, which are particularly important in the pathogenesis of neurological disease. It can also be subdivided by mode, and the most important mode is macroautophagy, and that is what I will be talking about when I talk about autophagy further on. And what the process of autophagy essentially involves is the engulfment of the targeted debris within the cell by a structure called the autophagosome. And this autophagosome then fuses with the lysosome, and the targets are digested in the lysosome. And the process of the fusion between the autophagosome and the lysosome is specifically disturbed in Vichy syndrome and related conditions. And that is, of course, a gross simplification because the autophagy pathway involves a large number of additional proteins, and I'm only showing this figure because all of these additional proteins and genes are potentially candidates for very similar disorders and to some extent have already been implicated in specific Mendelian disorders. And autophagy was initially a mechanism described in the 1960s, and initially was mainly of interest for cellular biologists. But then it became implicated in a non-specific way in a wide range of medical conditions, and in particular medical conditions which affect organs with post-mitotic cells, which have a high energy need, such as the cardiovascular system, the musculoskeletal system, or the nervous system. I also wanted to point out the dual role of autophagy in cancer. Autophagy is very important to prevent cancer by removing potentially toxic substrates from the cell. But within the tumor cell itself, autophagy actually plays a role in driving tumor growth. And until we described the group of EPG5-related disorders, autophagy had not been linked in a major way to Mendelian conditions. And after we had identified the causative gene, we continued characterizing the condition Vichy syndrome through large international collaborations. And I think that is a point which has been emphasized before, that particularly in rare disease research, it is very important to collaborate widely and on an international level. And one thing I also wanted to point out, if you look at the map, sadly, the global south seems to be rather underrepresented if you look at the distribution of patients. And that, I think, is something which hopefully is also something which will be addressed in future. And I wanted, that was the second series of Vichy syndrome patients with around 60 patients. And I wanted to highlight two aspects. One aspect is that some of these children have features which are not dissimilar to lysosomal storage disorders, and that is also reflected on the brain MRI appearance. But one aspect which I think is even more interesting is that if you look at the top row, you will see that these infants look developmentally actually quite normal. But if you look at the bottom row, you can see that these children actually do have quite significant neurological problems. And that emphasizes that this condition is not only a neurodevelopmental, but also a neurodegenerative disorder. And just to highlight the clinical features which illustrate that is the most extensive human multisystem disorder reported to date, I have highlighted in red the 7 features we consider diagnostic. If a patient has all 7 of these features, the likelihood to find a mutation in EPG5 is in excess of 95%. I've also highlighted in blue additional neurological features, in particular epilepsy, which is very common in these children, but also in black other multisystem features which illustrate that in these conditions virtually any organ in the body can be affected. And there's one other interesting aspect which I mentioned before, that you do find neurodevelopmental or developmental abnormalities in general, and on the background of the developmental abnormality, you may get a degenerative component. And that may not only affect the brain, but it is also a feature in any other organ, for example, in the heart, where you get cardiac defects, but then later in life you may get a cardiomyopathy. And that is reflected in an important role of autophagy, also in embryonic development. And we have taken our studies further with these conditions, and by now we have identified more than 200 patients with mutations in this gene. Most of them at the moment have truncating mutations highlighted in red, but we are finding an increasing number of missense mutations. And one observation we have made recently is that the condition called Witchy syndrome is actually only a small part of the mutational spectrum. And very interestingly, we have found a relatively milder part of the condition where much more common neurological disorder may be the main presenting features, such as Parkinson's disease, such as epilepsy, or milder neurodevelopmental presentations. And I think that is another point which applies to many rare diseases, that you start with a condition where only very few patients may have been recognized, but where the condition and the gene then becomes the cause of a much wider spectrum of disease. And one other observation I wanted to mention is that there also appear to be manifestations in heterozygous carrier of these mutations. Some of them have vitiligo and early onset cataracts, which seem to be minor forms of the hypopigmentation and the cataracts we see in Witchy syndrome patients. But more importantly, some heterozygous carriers also seem to have a higher frequency of certain cancers, and in particular movement disorders such as Parkinson's disease. And that corresponds to observations in other lysosomal storage disorders, for example, such as Gaucher's disease, where you can also get an increased Parkinson frequency in the heterozygous carriers. And of course, the main aim is to take from the bedside to the lab and then go back to the patients and offer therapies to the patients. I have to say that that part of the work was held up substantially by the pandemic, but we have recently made further progress. And we have an array of cellular and animal models, including C. elegans, Drosophila, and mouse. And that is the most exciting slide which I can show today, which is fresh off the press. That is a collaboration with my colleague Haiyan Zhu from UCL, with whom I have been working for the last 20 years on rare conditions. And she has recently designed antisense oligonucleotides for the recurrent EPG5 missense mutations, which results in an abnormal and a reduced normal transcript. And as a proof of principle, she has demonstrated that these, or one of these antisense oligonucleotides, can reduce the amount of the abnormal transcript and at the same time increase the amount of the normal transcript. So there's still a substantial amount of work to be done, but on the whole, these results are very promising. And when we did find Witchy syndrome, initially I did ask myself, is that just an extremely rare condition, or is it just representative of a larger group of conditions? And it turned out indeed to be the case that Witchy syndrome is just one of a major group of neurodevelopmental and neurological disorders. And although initially we had only described 6 conditions, by now there are almost 50 conditions which are linked to the autophagy pathway and to closely related intracellular trafficking pathways. And one molecular biological part of the pathway which has been particularly affected is the part which ensures the fusion between the autophagosomes and the lysosomes. And that has now been associated with a very wide range of neurological conditions, and the number of these conditions is still increasing. And all of these conditions are multi-system disorders. They have very prominent CSS involvement. They have variable neurological features. And the hallmark is that they start as neurodevelopmental disorders, but then become neurodegenerative disorders. And these conditions have a wide differential diagnosis. I just want to mention that the differential diagnosis is due to these conditions being linked in very similar biological pathways. And these pathways have very recognizable clinical signatures. That is something when we found the first patients of Vichy syndrome, I did a reverse search on a dysmorphology database, and all the conditions which come up here are due to mutations in genes which have now been linked either directly or indirectly to the autophagy pathway. And I want to conclude here on an aspect I find particularly important, that there is increasing evidence that congenital disorders of autophagy form a part of a lifetime continuum of disease. So neurodevelopmental disorders due to mutations in cellular trafficking and autophagy genes often feature early onset movement disorders such as Parkinson's disease. Vice versa, the genes implicated in these disorders have been identified as modifiers of neurodegenerative disorders. And lastly, there is an increasing number of primary autophagy genes which have been implicated in conditions like dementia, like amyotrophic lateral sclerosis. And I think one condition I've highlighted is the condition due to FIC mutations in FIC4, which causes in the heterozygous state ALS, but in the recessive state a neurodevelopmental disorder similar to Vichy syndrome. So to finish here, the genes implicated in autophagy disorder prevent certain cellular housekeeping mechanisms which usually ensure good cellular health and shift the balance toward mechanisms which promote cellular aging. So to some extent, I consider these conditions also disorders of premature aging. And I just wanted to highlight one aspect, because as clinicians we focus very much on disease, but I think what's also important is that these conditions also illustrate mechanisms which are very important for good health. So for example, autophagy is probably also the mechanism why fasting is good for your health. So I skip my last slides because I'm running late. And I just want to thank my clinical team, a large number of collaborators, and in particular also the charities who have supported this research. Thank you.